Various studies in science and in the field of biology have referred to genetics as the study of variations and heredity. You have known in probably earlier years of study that heredity is a transmission of characteristics from parents to offsprings, and that's particular understanding that is clear. Now, variation would refer to the differences that exist in individual populations. That's why we have a wide range of differences in terms of characteristics, features, and traits among organisms, even within a specific species, and that's important. And so that introduces an interesting idea of genetics, having a drive to investigate into what the factors behind these variations we see are, and what is causing those variations to be transferred from the parent to the offsprings. And so that makes genetics very much interesting. What this means is that fundamentally, you would have to understand the modalities or the processes or the specific mechanism through which those variations are transferred from the parent to the offsprings. And it's been said time and again that they are transferred through hereditary units called genes. Today's video tutorial is going to enlighten you on every concept or everything that exists as a body of idea about gene. And so this tutorial is about the modern concept of gene, enlightening you about everything that you need to know about genes. So before we continue with the tutorial, kindly subscribe to this channel and share this video for others to be able to enjoy. My name is Michael Ampayeboa, and we're having a drive through the modern concept of gene. And so the idea of gene was first proposed by Mendel, Johann Gregor Mendel, in the year 1865. In this particular extent, he did not refer to it as a gene, but in his studies about inheritance of acquired characteristics, he called this factor. So he said some factors were responsible for the transmission of characteristics from parent to offsprings. It was later on in 1909 that one scientist called Johansen was able to propose the name gene for the term. So the idea was proposed, the idea was discovered by Mendel, who then suspected that certain factors were responsible for transmission of characteristics. And then later on in 1909, Johansen gave it the name gene. Today, as we know in the world of science and biology, a gene is referred to as the basic and fundamental unit of heredity and, for that matter, life. What it's also important to notice that genes control both specific structural and functional features of an organism. And that is to say that genes are able to control the structure of specific functions of cells and, by extension, the entire organism in that regard. Now, genes are present in every cell and organism for that matter. Chemically, we know that genes consist of specific sequences of DNA building blocks. And the building blocks of DNA, that is to say that the subunits or subcomponents of DNA, which are called the nucleotides. You know that a lot of nucleotide-based pairs would have to come together to form a DNA, right? So the building blocks of DNA called nucleotide, and these nucleotide sequences come together to form a gene. Each nucleotide, as we know, is made up of a pentose sugar, nitrogenous base, and a phosphoric acid. On the average, a gene consists of about 1,500 nucleotide base pairs, and that information is particularly important. The strands of DNA into which genes occur are called chromosomes, and so genes, DNA is packaged into a specific unit called a chromosome. And on chromosomes, we will be able to locate a lot of genes on the chromosome. And so from the lowest order to the highest order, you move from gene, then you move from DNA, then you move to genes, then you move to chromosomes. If you even want to start from nucleotides, then you move from nucleotides to DNA to genes and then to full chromosomes. And that's important. Now, each gene is going to serve as a blueprint for the synthesis of enzymes and other proteins. That is to say that a gene will become the particular programming factor that would help in the expression of the individual characteristics or the code that varies. So for example, if an organism wants to synthesize a certain protein, there is a particular code that exists in a gene that would be able to serve as the model for which that protein would be synthesized. A single gene may occur in several forms, and the various forms or the alternatives of a gene that we have are called alleles. So alleles are then referred to as the alternative forms of a gene. So it's a single gene. So for a gene for height exists in several forms, which will be that the form that is responsible for shortness and the form that's responsible for tallness. Okay, so the variation of the gene for height, the form of the gene for height that's responsible for shortness is an allele. The one that's responsible for tallness is also an allele. But however, the two come together to form the functional unit of the gene, and that's important. So generally, a gene has two forms or two alleles, and we have two particular known types of alleles that we have: the dominant allele and the recessive allele. Now, this is just like the cat and mice situation. You know that the cat is always dominating over the mouse, and the cat will always win over the mouse. We think that the dominant gene is the cat in this situation, and the recessive gene is the mouse in this situation. So whenever the cat is away, the mouse can play, but if the cat is around, the mice cannot actually show or express themselves. So what this means is that whenever a dominant gene is present, the particular feature or characteristics that is controlled by the recessive gene would not show. It is only the feature that is controlled by the dominant gene that is going to be expressed in the phenotype. By phenotype, we mean the outward characteristics or the express feature that we are taking into consideration. So a recessive gene is more powerful. A dominant gene is more powerful. A recessive gene is less powerful. A dominant gene will always express and prevent the recessive gene from expressing itself, and that is a particularly important information. Now, sometimes, even though we know that genes normally exist in two forms, which are the two alleles, sometimes there are some characters that are controlled by genes that are expressed in multiple forms. Okay, they are expressed in multiple forms, and that is a situation that is called multiple alleles. A typical example is blood grouping in humans. You can have the allele A, which is responsible for blood group A. You have the allele B, which is responsible for blood group B. You have the allele AB, and you have the allele O, which is responsible for blood group O. So you have three in this particular instance: A, B, and then O. In this particular instance, we have three different alleles, and this is called multiple alleles. It's an instance of multiple allelism. Now, even though we talked about the idea of dominance and recessiveness in terms of genes, sometimes it's not always the case that genes present themselves as the alleles in a gene will be, one of them will be dominant and the other is going to be recessive. Sometimes they equally express themselves, and that instance is known as codominance. They are equally dominant and all of them are going to be expressed equally. So for example, in cows, if the allele for black fur is more dominant over the allele for white fur, then anytime the allele for black fur is present, the cow that is produced is going to be black, right? But an instance of codominance, you would find the cow being black and white because all of those genes are equally expressive and they're able to express themselves equally in a concept known as codominance. Genes can also replicate and produce copies of themselves. The expressions of genes is influenced by a lot of environmental factors. So even though genes contain the blueprints for producing proteins and for that matter, controlling characteristics and features of organisms, environmental factors have a great influence on the expressivity of the genes. The total set of genes that exist within an organism is called a genome. So that's important. If you want to consider all the genes that control all the various characteristics within an organism, you are referring to the genome of that particular organism. So for example, in humans, we have 46 chromosomes that contain a lot of genes. If you're taking all of these genes there are in humans into consideration, then you are considering the genome of humans. Genes may also vary in makeup from one person to another, and that's a very interesting concept. Each gene occupies a position on a chromosome called the locus. A plural is the loci. So if you take a chromosome, there are specific points, there are specific places within the chromosome that you're going to find genes. And those particular points on the chromosome where genes have a high probability of occurring, those places are called loci. The singular is locus, right? Genes are also arranged into a single linear chromosome, and in most of the instances that we find in humans. The alteration in the number and arrangement of genes is what we call mutation. So sometimes the number of genes that are supposed to be present is going to be reduced or be increased more than necessary or less than required. Or the forms of those genes are going to be changed, are going to be altered through a lot of factors, be it environmental or genetic factors or any other factors relating to cell biology. That particular instance or concept is called mutation. So mutation is a change in number and arrangement of genes, and this can cause a lot of problems of clinical importance. Changes that occur due to mutation in germline cells have the potential of being transferred to the next generation. So by germline germ cells, what I mean is that if a certain mutation exists on genes that are contained in a sex cell, then that particular mutation would be passed on to the offspring that is going to be produced by that particular individual. Mutations that affect somatic cells may result in various types of cancers, and that is also of very crucial clinical importance. So if the mutation is found in sex cells, it will be passed on to the next generation. If it's not in sex cell but in somatic cell, which body cells, then it's not going to be passed on to the next generation or the offspring, but it results in serious degrees of cancer, which are of clinical importance. A lot of genes that we have that are present on chromosomes are inherited together. So most of the times, by that particular combination, certain genes are always going to be inherited together, and there is very little probability of they being passed on separately. Always they are going to be inherited together, and such genes are called linked genes. So for example, you ever heard of the anecdote that says that people who are left-handed are very quick-tempered? That particularly means, if that statement is true and empirically verified, what that will mean is that the genes that control left-handedness and the gene that controls quick temperament are linked genes, and they are always inherited together and not separated. That is what the meaning of linked genes are. Now, sometimes you can have a lot of genes that interact to produce a particular trait, and this particular concept is called interaction of genes or gene interactions. Now, on other instances as well, you find that a single pair of gene is producing two or more characters, and this is known as pleiotropy or pleiotropism. We know that one gene, one protein. That's a hypothesis that is very, very much interesting and prominent. We expect that every gene controls one trait. However, we are also exposed to certain genes that have the possibility of controlling two or more characters, and those genes exist in a concept called pleiotropy or pleiotropism. So in pleiotropy, a single gene controls two or more traits or characteristics. Now, the transfer of alleles of genes is able to occur from one population to another, and this is called gene flow, and this particularly can lead to changes in the gene frequency. So if individuals belonging to population A are able to interbreed with individuals belonging to population B, then there's going to be gene flow, and that is going to cause changes in the frequencies. how often we are seeing the genes, a certain gene expressing itself within a population, because those changes do occur. Now, the number of genes in a particular cell may be increased by polyploidy or hyperpolyploidy, or be decreased by hypoploidy. Now, what does this mean? In polyploidy or hyperpolyploidy, you have the particular set of genes, the amount of genes that we are expecting to see in an organism increasing by certain folds. This is something that we will consider later on in another video. In hypoploidy, you realize that there is a reduction in the expected number of genes within that particular organism. Inbreeding is a phenomenon in which organisms belonging to the same population are able to breed with themselves, and this is something that can lead to homozygosity. And by homozygosity, we mean that alleles that are of the same type meeting together and expressing within an organism. Outbreeding, which is one population mating with another population and exchanging genes and cross-fertilizing, that will lead to what we call heterozygosity. And that has been proposed as the most ideal way of reproduction because it leads to a lot of variation, resulting in fitness within the offspring that will be produced, and the gene pool is going to constitute a lot of variability or different combinations of gene, and that's contribute to fitness. There's going to be more understanding of this in the other videos that are within this playlist. Now, the fine structure of genes has revealed that a gene consists of several parts. So when you take a single gene, you can have different points of the genes that are responsible for certain factors. Okay, so we have the cistron, we have the recon, and then you have the muton. The cistron refers to a portion of DNA or a gene that specifies a polypeptide chain. So the part of the gene that can code for protein is the cistron. The recon is the smallest unit of DNA that is capable for recombination. The muton is the smallest unit of DNA that's capable for undergoing mutation, right? Now, some genes are naturally split such that they contain some sequences which do not code for proteins, and also sequences that code for proteins. The sequences of such genes that are split, that do not code for protein, those non-coding sequences of those genes are called introns, and the coding sequences of those genes are called exons. Genes having one intron is called a monointron gene. So if a gene consists of just one intron, it's a monointron gene. And if it contains more than one intron, it is called a polyintron gene. And that is really important, or a multi-intron gene in some instances. And a typical example of such gene is the rat muzzle X-actin gene, which has 6 introns. That's a very interesting phenomenon. Now, there are also certain genes which code for more than one protein, and such genes are called overlapping genes because they are responsible for coding for multiple number of proteins and overlapping genes. Now, the overlapping genes share the same sequences. So, for example, in certain bacteriophage, you have gene E lying entirely within a gene D1, and they are translated in different reading frameworks such that at different instances, they result in producing different types of proteins. There are also genes which move from one location to another on a chromosome. So they are jumping genes, right? They move from one point of the chromosome, one locus of a chromosome, to another locus of a chromosome. And such genes are referred to as jumping genes or transposons. A gene that produces 2 proteins simultaneously from a long transcript by changing the endpoint of protein synthesis is known as a nested gene. And the entire coding sequence of such a gene lies between the start codon and the terminating codon of a larger external gene. Now, that means that the entire coding sequence are present in other genes. That's a very interesting concept. The genes that also have lost the ability to perform function. So there are some genes who have lost the ability to perform their functions due to mutations, and they are called pseudogenes. They look like normal genes. That means that structurally they are genes, but functionally they are not performing anything or not corresponding to the production of proteins. They do not, they look like normal genes, but they do not express RNA or protein. They are often termed as non-functional DNA, or something that is also regarded as junk genes. Now, genes that lack a well-defined exon or intron sequence, that is to say that the exon in one instance might be the intron in another instance, is termed as a complex gene, and such complex genes exist in a lot of instances. These genes manifest excessive rearrangement in the sequence of mRNA. That is to say that before post-transcriptional modifications, a lot of changes can occur within such genes. Now, genes that cause cancer are referred to as oncogenes, and oncogenes are activated by what we call proto-oncogenes. Genes that code for proteins which act as transcription factors, enhancing the rate at which certain DNA sequences are transcribed, are also termed as homeotic genes. And it is important to note that homeotic genes play a key role in the early development and differentiation in embryonic tissue in eukaryotic organisms. These genes were first identified in Drosophila. Now, also, the group of genes showing similarity with each other is termed as family of genes, and they arise from duplication. Now, all genes in a family may occur on the same locus, and that is to say that, for example, 5 members of growth hormone gene family are clustered around chromosome 17 in humans, and that's a very typical, a classic example of family of genes. Now, gene families provide information on how new genes arise and diversify. Another interesting concept to note is the genetic code, and the genetic code refers to the linear relationship between amino acids and polypeptide change and a triplet of DNA or RNA. But this is a concept that is worth dedicating a full section, and we consider it in the subsequent video. Thank you very much for enjoying this lesson. My name is Michael Ampayeboa, and I have been your partner in today's tutorial. Don't forget to subscribe to this channel, hit on the notification icon, the bell icon, and you get notified anytime I post a new video. And also share this video to your friends.